Hi, guys. Okay, so we're going to finish up chapter 3 with this second part here. So let me go down in the corner. Now, so we're going to talk in this course in developmental psych, we talk a lot about normal development, norms, what's typical, what do we see kind of a pattern of. Not everything fits the norm, but in normal development, we all get the same number of chromosomes, 46, those same types of genes lining up on those chromosomes in the same places, the same 4 bases kind of making, that's my chair moving, by the way, same 4 bases making up the same kinds of proteins. So we all, as human beings, me, you, everybody listening to this, everybody you know, share more than 99% of our genetic makeup. We even share about 25% with rice. So I always think that's interesting. 25%. And, people are like, what? What do you share with rice? So all animals, plants, and fungi share an ancestor that lived about 1.6 billion years ago. So through evolution, if we go back to the very beginning, we still have some of that very early genetic makeup. Evolution does not do away with, it just builds complexity over time. And so the parts that worked in the beginning have remained, and we haven't lost it. But we use parts of it for different things, more advanced, more complex, and so forth. But anyway, more shared than not. And as human beings with 99% of our genetic makeup shared, we are much more similar than we are different. But we are unique. Unless we're identical twins, we don't look exactly alike. Even identical twins can change. We'll look at that. So, what is it that makes us unique in the light of all of this sameness? So let me get rid of myself here. The number one way is alleles, and we've talked about alleles. So that's one of the options for a gene. So as mom and dad's chromosomes line up next to each other, as babies takes the chromosomes from mom, they line up next to each other with all the genes lining up in similar places. You could have one for blue eyes, one for brown eyes, one for curly hair, one for straight hair. So what that option is are different alleles. We usually get 2 copies of every gene, and they can be slightly different, so the code is different. The differences impact what proteins are made or change how much of that protein is made, thereby impacting the eye color of the life made by the 2 or whatever it is that's being made. So alleles, the options whatever you get from mom and dad, and then how they integrate together make a difference. The second thing is mutations. When cells duplicate, the copy is usually spot on, just like the original, but not always. So, cells continually things duplicate to, in this process of development. And sometimes there's slight changes in those base pairs. It's like the copy machine just doesn't get it exactly right all the time. There can be a swapping, deleting, duplicating. We'll look at that in a second. So these changes can get passed down. And in new generations, more mutations can be made. So one way we can look at this, like blonde hair was created through a mutation. It's just one little letter swap in base pairs. As humans moved north in the early days, dark hair and skin weren't as necessary for protection from the sun. So when a mutation occurred, making hair or skin lighter, it sometimes stuck and got passed on because it wasn't bad for the person. It wasn't, a lot of things created by survival of the fittest. It was something that could stick and so then gradually be passed on. A SNP, SNP, or single nucleotide polymorphism, is the actual change in the code at a particular place and refers to a single swapping of letters in that code. So, we used to think this was what impacted most changes or differences between people. It's not. There's not as many just single letter swaps, like single base pair swap or something, but there are some, and blonde hair happens to be one. So if you look down here, types of mutations, and you can see these are the different, this is the copying of one chromosome to another, or one cell to another. And so there's a deletion here, a duplication of genes here, there's an inversion, see how it's like 1 up here, 2 here, and then it inverts here, insertion, translocation, so it gets inserted into a different, this little piece gets inserted in the middle of this, or this piece gets inserted onto the top or bottom of this. So these things can happen during duplication. And they a lot of times create, not problems, just little differences in the way we show up in the world. And sometimes they stick. And especially if they do end up to create something helpful or beneficial to the species. This is how, most of the things this is how a lot of the evolution has taken place to create things that have benefited us. They stick from a mutation. So here's another one. The book doesn't talk about this at all, recombination or crossing over. But I think this is really interesting and really important to know. Before germ cells, which are those reproductive cells, split and lose half their DNA, they still have 46 chromosomes. So the germ cell has all 46. And so before it splits, so this makes sense, before it splits, it duplicates itself. So then it has twice as many, a copy of each one. So then it lines up. You can see this over here. There's actually, so this is, two, like a mom and dad, and this is another two. So they've each duplicated themselves. So we end up with twice as many, making identical copies of every chromosome. Then there are 2 copies of moms and 2 copies of dads, and they all line up next to each other. But before they split, the chromosomes exchange. If you look over here, the cell with 46 chromosomes, 2 unique copies of each. The chromosomes exchange bits of themselves. So this is what I find really interesting. They make a recombination. So each of the 4 copies of chromosomes become part mom and part dad. It's not all mom, all dad. It's actually a little bit of both. So it gets all kind of, it blends and mixes up a little bit, and they become very uniquely combined. And this is why 2 children, at different times that aren't identical could never be exactly the same. They split into 4 cells then. These germ cells, they make 4 because each one is only going to have 23. It's going to be a sperm or an egg, and each is completely original. You can see how the color is a little bit different, showing some of the recombination. The genetic material in each of these looks different than the DNA of mom or dad's. If these cells have the opportunity to become fertilized and make a new person with a genetic code, it's unique from any other because of this recombination. So we go from 2 identical copies to 2 unique copies, and then it splits off into 4. So I think this is one of the big ways we end up unique. And this happens to all of us. This isn't a mutation or anything. This is the way our reproductive cells are created. Okay, so then the last thing, copy number variations. Though we typically get 2 copies of every gene, sometimes we get more or less. Though like most variations in the genome, copy number variations cause little that is outwardly noticeable. Some play a role in disease and healing. And there's a lot of research still being done. Because as I said earlier, we used to think that SNPs, that single switch, used to be... we used to think that it accounted for most differences, but now we know it's this copy number variation. So if you look over here, as this gets duplicated, here we see 1 copy of C of this gene. Here there's 2. There's 1, and there's okay, we have I'm sorry. We have 1 of each, okay, C and C. In this duplication of the cell, then 1 of those got deleted. And there's only 1 copy of C. In this one, there's 3 copies of C. So of course, any change in a code, just like a computer code, any little change in it is going to make a change in the final product. So this is true for babies too. Any little minor change in that code, deleting, duplicating, swapping, inverting, whatever it might be, is going to create a change in our final product baby. Okay. So this is a lot of information, I know. I'm sorry. Just hang with me here if you are so interested. So gene expression patterns, so once these, the chromosomes have lined up and the genes have lined up next to each other, and now moms and dads are going to combine in some way to figure out what it's going to look like in baby, this can happen in different ways. So additive, a baby's trait is a blend of those two alleles. And so hair texture and skin color are a lot like this. So if you have wavy haired mom and straight haired dad, those blend like you're mixing paint. And you get, if you have curly haired and straight hair, you're going to get a little bit of wave, a little more or less, somewhere on that continuum that's going to be. Same with skin color. Dark-skinned mom, light-skinned dad, they're going to mix like mixing paint color, and you may get something of the two combined. So that's one potential way. And then we have dominant recessive. And I've tried to explain more than you're going to get in the book here, so it actually makes some sense. A lot of this, again, you teach what you need to learn, has gone into the learning for myself, research that I've done. So when one allele is dominant, and so remember, an allele is whatever copy of a particular gene that mom or dad have. When one is dominant and the other, this dominant recessive is when one is dominant and one is recessive. So an allele is considered dominant because of how it codes for proteins. So some proteins are dominant. They win out over their fellow proteins, just like dominant athletes win against their competitors. So some just happen to be stronger. The simplest situation of dominant and recessive alleles is if one allele makes a broken protein. So when this happens, the working protein is usually dominant. The broken protein doesn't do anything, so the working protein wins out. There's different ways. This is complicated and in research, and probably if you really were interested in knowing why some are dominant and some are recessive, I welcome you to go do a lot more research with that. So if a child, when that combination is made, gets one of each, a dominant and a recessive, the dominant trait will express most of the time. So if you've ever done any of this in any of your classes, you've talked about eyes probably. So blue, and you'll see it has the little b, and that means it's recessive. Blue is recessive and brown is dominant, big B. So if you get a big B from mom, brown from mom, and so B from mom and dad equals B. That wasn't written very well, but both give you the big B. Oh, I'm sorry. I'm not even reading my stuff right. You get a big B from mom and dad, you're going to get brown eyes. If you get a big B from mom and a little b from dad, you're going to get brown eyes. A little b from mom and a little b from dad is the only way you get a blue-eyed baby. You have to have two of the recessive in order for the recessive to express. If the child gets one big B, one little b, they still carry that little b, so they can pass it on. So if they marry someone that has two little b's, it's possible that when those two come together, baby may end up with two b's. That's a potential. So looking at red hair is a recessive trait. A protein called MC1R has the job of getting rid of red pigment. So if you get a gene where that isn't working for some reason, where there's no MC1R, you get a buildup of red pigment and end up with red hair. Now, obviously, there are some cultures in which red hair actually looks dominant. There are cultures in which blue eyes are dominant. And that just has happened over time in evolution. A lot of blue-eyed people mating with blue-eyed people, or red-haired people mating with red-haired people. So it's not, keep in mind, recessive and dominant doesn't mean like in your family, what's the dominant trait. This is the way, there are dominant proteins and recessive protein, dominant proteins and that are going to be stronger across the board. In the genetics of all human beings and how they mix. But yeah, you can get a culture with a lot of blue eyes if blue eyes and blue eyes and blue eyes and blue eyes have stuck around for a long time. So like in Norwegian cultures, you might see that. So other traits, there's a lot. If you've been in like other psychology classes, you might have looked like the hitchhiker thumb. can never do this on the thing. And the straight thumb, and talked about that. And I happen to have one of each, which I don't guess you're really supposed to have. It's kind of my superpower. I don't know. It's really, this is thinking that all of these things are sort of based on single genes, and that's, it's not usually as simple as that. So it can be a lot more complicated. But here's a couple of other interesting ones that probably have a lot more to do with it, but we can kind of throw them out as ideas. The dominant version, so bent pinky, if you look at your pinky, if the top segment bends towards your other finger, that's dominant. And mine actually does, I realize. I've always wondered why as I think about that, I used to wonder when I was young, why it's bent like that. So mid-digital hair, this is on your fingers too. People lacking hair in the middle segments of the fingers have two recessive versions of that gene. So hair is obviously dominant on the fingers. So that's just another little one to think about. All right, I'm going to hide myself here. I wish I could put myself in a different corner. Hazel eyes, because I'll have students ask about this. So just a little more information on the eye deal. Brown, green, and blue eye color comes from a pigment called melanin. Brown eyes have a lot of melanin in the iris. Green eyes have less, and blue eyes have little or no pigment. So there's two genes, BAY2 and GAY, work together to make brown, green, or blue eyes. One form of BAY2 makes a lot of melanin, so B, while the other form makes only a little. One form of GAY makes some melanin, while the other makes only a little. So this is big green, and then this is blue, brown. If you have B, you get brown eyes. G, but no B, you get green eyes. And if you only have the little b, then you get blue eyes. Hazel eyes, still not known for sure. Honestly, they don't really know why people get hazel eyes. There's not a lot of research being done on it, I don't think, but there is some. Most likely, though, one answer is that hazel eyes simply have more melanin than green, but less than brown. Maybe different genes are actually involved. There may be like a hay for hazel or something, and maybe hay is a bit like BAY2 and GAY in that it comes in two forms, one that has enough melanin for hazel and one that makes little or no melanin for blue. So it's going to be a combination, though, of, again, the way mom and dad's alleles combine together and what those certain genes are, how much melanin they make. That isn't known as an exact science yet. So, but kind of interesting. You can look into that more if you like. All right, let's move on. So when things go wrong, the genetic of disorders, it's really amazing how much the human body can get right what it does. But there are times when it doesn't, when things go wrong. And some of that, like mutations or copy number variations, those aren't really things, they are going wrong, but not going wrong in a way that causes necessarily, maybe sometimes, but not usually, problems. But there are things that can. So after recombination, so that's, remember when the germ cells copy and they recombine, cells divide into 4 individual reproductive cells. When this happens, sometimes the chromosomes are incorrectly distributed. So this is when baby is going to get the wrong number of chromosomes, and this can cause problems. So one cell might get 2 copies of a chromosome, another gets none, some may be flipped. If a cell with too many or too few chromosomes takes part in fertilization, the zygote gets too many or too few chromosomes as well. So often fatal to the zygote. This is, when we have miscarriages, because the body realizes this wasn't done right, that a child cannot live with too many or too few, because if you think about it, there's not enough genetic material to create what needs to be created in order to develop normally. We need, all of that goes into our code. Now, the information on the genes, how it combines and so forth is a little bit different, but the actual number of chromosomes we have is very vital. Sometimes a baby is born with an extra or missing autosome and has a disorder of some type. So, different chromosomal disorders. Trisomy 21 or Down syndrome, a child born with 3 copies of gene 21. So if you look here on number 21, these are all of them. There's 3 copies. So that's when a child ends up with Down syndrome. During the recombination, something was duplicated or added. Monosomy X or Turner syndrome, a female child is born with only 1 X chromosome. So a female should have 2 X's, they only have 1. The genetic material that is missing causes developmental problems before and after birth. So that can even happen. Some dominant and recessive disorders. An individual can develop a disorder while carrying only 1 copy of the gene marked with the disorder. So if one is normal, but one has a disorder on it, if the carrier gene if the one with the disorder is dominant, therefore it's more influential, they will have that disorder. So Huntington's disease is a brain disorder causing a lack of movement control and cognitive and emotional problems, and it's a dominant disorder. It is caused by a mutation in the HTT gene. So if this happens, you only need 1 and on the dominant gene. And then, it's dominant. And so if it shows up there, then the baby will have it. Disorders dictated by a single gene, of which 7,000 are known, are most often on the dominant gene and therefore expressed. For a disorder to be on a recessive gene and be expressed, the disorder would have to be on both copies of the gene. So both parents would have to have the disorder. So sickle cell anemia is a big one, and I think just a good example to explain this, but it's also kind of an interesting one. In order to have sickle cell anemia, you have to have it on both genes. 2 carriers on autosomal chromosome. Remember, autosomal is every one except the sex chromosome. So all 22 pairs are all called autosomal. Chromosome 11, it's the hemoglobin gene. So it affects the red blood cells, which use a protein called hemoglobin to transport oxygen from the lungs to the rest of the body. Red blood cells are usually round and flexible, you've seen maybe pictures of them, and can travel freely through narrow blood vessels. A mutation on gene 11 that causes the cells to be rigid and have a concave shape. This is what sickle cell anemia, it actually looks like a sickle. If you think about a tool, that's called a sickle. These cells get stuck in the blood. They're like a shape that then doesn't move smoothly through. It gets stuck, and they can't travel through the vessels. They can't transport oxygen effectively and cause pain and damage to the organs. It is most common among people from Africa, India, the Caribbean, and Middle East, and the Mediterranean. The reason that might be for that is that it actually has to have 1 copy of this is protective. Makes the carrier resistant to malaria, and malaria is most common in these places. And so through evolution and sort of survival of the fittest, it behooved people to have 1 copy of this, so it didn't die out altogether. And yet if you get 2, which in a population in a specific culture, if you are mating or marrying within that culture and there, then there can be the problem of having sickle cell anemia. Which is why, this is the kind of reason why it's sometimes good for people of the same culture, same kind of genetic culture, to have genetic counseling to check for these kinds of things, because there are some disorders that are more common among particular cultures than others, and this being one within these populations. So if 2 people were going to have a baby and they were both African or Indian, Caribbean, Middle Eastern, or any combination of those where this is more prevalent, then they might want to find out, because 2 copies of it could end up in sickle cell anemia. And you can be a carrier and it not express. All right, so X-linked disorders. Some disorders are X-linked, which means they're on the X chromosome, rather than an autosome. More men display it. Now that's interesting, because the X chromosome is sort of the female chromosome, comes from the female. Females have 2 of those. yet it's more men display the symptoms. So why? Because a female has two X chromosomes, and if you remember our picture earlier on of the X and the Y, you might remember that the X is much larger, much more dominant, has a lot more, it has a lot more genetic information on it. Two X's, the normal one may cancel out the other, but for men who only have one X, the Y is not going to cancel out the disorder on the X, and so it will express. X-linked disorders can include colorblindness, Fragile X syndrome, hemophilia. So genetics or environment, and then of course this makes us different too. If we look at our, when we combine environment and genetics. Alcoholism, for example, genes are 50% responsible for someone's tendency. That does not mean you are definitely an alcoholic. There may be a tendency to become addicted to alcohol. 2014 study by Scripps Research Institute located a gene that very well might increase risk. Then there's some personality traits too. Quick temper, thrill-seeking, inhibition, which may all make sense in one way if you look at them separately. But then environment. A person might have a genetic pull toward alcohol, but depending on their family, culture, peers, they may or not be triggered for gene expression. So this is why like college is a really big time for this triggering, quite honestly. If someone has that predisposition or that tendency genetically, and they put themselves into, say, a college environment where there's a lot of partying going on, a lot of drinking going on, it can be really hard for those people with this to not get triggered and then end up having a really hard time breaking that. I saw that a lot in college. There are people that could go to college and drink themselves silly for 4 years and then stop and never have a problem with it. And then there are those who could go and do that and they end up with a problem with it. Schizophrenia is another one. Schizophrenia, a lot of the mental health disorders, no one gene has been found to be a hallmark of it. Several genes could increase risk. There's a heritable, which means inherited quality. 10% of those with a direct family member also develop it. If one twin has it, the other has a 50% chance of developing it. But there are environmental pressures, so it has an environmental aspect to it too. Prenatal malnutrition, virus exposure, childhood trauma have all been linked to increased risk of it triggering and becoming expressed. Infertility. About 12% of couples find themselves unable to get pregnant. There are choices. Assisted reproductive technology. Now, this includes all techniques used to help with conception. Assisted reproductive technology. So anything that could help. Drugs. Clomid is probably the most common. If a woman cannot get pregnant, that may be the first recourse. Try Clomid. So Clomid stimulates the hormones in the brain, signaling them to develop and release an egg. Gonadotropin acts directly upon the ovaries to produce and release an egg. To do this, it's more of a direct route. It acts directly on the ovaries, not on the hormones in the brain. But it requires daily injections and therefore is more expensive. So Clomid is usually a first go try at this. And drugs are usually a first try. And then in vitro fertilization is another choice. Eggs are removed from the ovaries, placed in a lab dish with many sperm for hopeful fertilization. If an ova is fertilized, it is inserted into the uterus. Almost half of these babies are twins or triplets, and often are premature and are low birth weight. Genetic issues are slightly more common. Twins and triplets in general are going to be way less typically. So, you have same resources, but 2, 3, 4, 5, 6, 7, 8, 9, whatever many to feed and give resources to. Intracytoplasmic sperm injection is really a part of in vitro fertilization. It's not something in and of itself. It's another form of art used to increase the success rate of IVF. In this procedure, a single sperm is injected directly into an egg. Instead of kind of just put together and hoping that things happen. And it is successfully fertilized, transferred to the uterus. Again, it's probably going to be much more, more expensive, more cost, more things. And none of this is necessarily cheap, but these are options for couples. Genetic counseling and testing. gives people genetic counseling offers couples the opportunity to learn more about risks, just to be counseled on them. Testing gives them the knowledge of personal risks, looking at what you might be a carrier for and so forth. Testing is useful for individuals with direct family members who have or had a genetic disorder, couples who have had several miscarriages, so there might be something genetically that needs to be considered. Infertile couples from the same ethnic group, I talked about that. Women over 35 and men over 40, especially the women over 35, remember what I was talking about, that we only get 200 to 400 in a lifetime. And the older we are, the older they are. And so genetic problems can happen more often. So that's the end of chapter 3. It's been 30 minutes for this. I am going to play the video now. And if you want to watch it, sorry, I actually didn't mean to stop that. Anyway, I'm going to play this video. If you want to watch it here and now, and I might talk a little bit about it afterwards, great. If you don't, it's on D2L, or you can come back to it later. I do find this very interesting. I have to pause for just a moment to resize the screen.
Identical twins are genetic carbon copies. The DNA sequences of their genes are exactly the same. Yet physically, identical twins become increasingly different over time. Why is this so? Genes provide the instructions for the development and maintenance of the body. Yet a second set of instructions, known as the epigenome, interacts with DNA to activate or suppress the expression of particular genes. Certain chemicals, known as epigenetic tags, turn genes off or on without changing the underlying genetic code. During fertilization, one set of chromosomes from dad and one set of chromosomes from mum are combined to form an embryo. Epigenetic tags are erased from mum and dad's chromosomes during the first days after fertilization. However, on some genes, the tags remain. These are known as imprinted genes. Identical twins result when a single embryo splits in two. Each embryo has the same genome and the same epigenome. As the embryos develop, cells differentiate into distinct types such as bone, muscle, and skin. As each type specializes, epigenetic tags activate and silence specific genes, leading the cells to specialize further. Each cell type gradually takes on a unique epigenetic profile. Because the twins share the same environment, their epigenomes are very similar at birth and through the early years of life. As the twins age, their environments begin to differ. Their genomes remain the same, but signals from the environment act on the twins' epigenomes to activate and silence different genes. Diet, for example, is an environmental factor that can have an effect on the epigenome. Differences in physical activity can also cause epigenetic differences between the twins. Exposure to toxins can influence the epigenome. Stress also plays a role in shaping the epigenome. By the time the twins are well into adulthood, their epigenomes, thus gene expression patterns, are very different, making each twin unique.
Okay, so I find that really interesting. This is an area that is having a lot of research done, epigenetics. So it's not just how does our environment, which we'll talk a lot about in 0 to 2 and as we go on, not just like how does our immediate environment when we're already alive impact our belief systems and our ideas and our perceptions, yes, but even at the genetic level, the things that our parents and our grandparents have been through will impact the epigenetic tags on their chromosomes. So there's research that even shows that like people who with PTSD, that that's going to affect their epigenetic tags. That's going to have an impact on their genetic coding, what genes are turned on and off. And these epigenetic tags are actually little chemical receptors on the genes that turn them on and off. And so, say PTSD in a grandparent, and that epigenetic tag, if it's not dealt with and worked with and changed over time, then that epigenetic tag can be transferred to a parent and to a grandchild. And so the way that our parents and grandparents and have dealt with life and have interacted with life can impact the way our genes end up looking. And the beauty about the neuroplasticity and plasticity is that we can take what we're kind of born with, though. We work with it the best we can. We work to change the patterns of our brains in the ways that we need to impact our body in healthy ways. And then we create a different epigenetic pattern for our children. But they're still going to carry things from grandchildren and grandparents. And so this is, it's fascinating information that I think you need to keep in mind as we move on. So that is going to be the end of chapter 3. Until next time.